Hi there. Alright, today we'll be talking about some various aspects of the cell. In particular, we're going to be focusing on two main things of cell biology. That is cell transport, so how cells get stuff into and out of the cell, and then metabolism. So it's kind of an abstract discussion of energy and how cells need energy. But we're going to start with cellular transport. In order to understand cell transport, we need to understand and revisit what we know about the membrane. So remember, the membrane is a double layer of phospholipids that separate the interior portion of the cell from the outside portion of the cell. So phospholipid, you'll recall, has two parts to it: a hydrophilic head. So the phosphate head is hydrophilic, and the hydrophobic fatty acid tails, are hydrophobic. All right, so this molecule by itself actually is a little bit confused when it comes to water. Half of it is attracted to the water, and half of it's repelled by the water. But when you put a bunch of them together, they will spontaneously form this bilayer, where the phosphate heads are oriented towards the interior, where there's water, and towards the exterior, where there's water. And in between are the fatty acid tails. Right? And it's these fatty acid tails that are the boundary layer that separates the movement of particles, that prevents the movement of particles from the interior and exterior of the cell. Right? So you got to think of the inside of the cell as water, and the outside of the cell as water, and the barrier is that oil fence. Right? So let me explain what that's going to look like here. So you have a phosphate head, fatty acid tail, and you'll have a double layer of this, right? That's because the inside of the cell, the cytoplasm, is basically water, and the exterior as water, right? But here in the middle we have our nonpolar fatty acid tails, and that's the barrier between the inside and outside of the cell. So what kind of stuff is going to be dissolved down here inside the cell? Well, because water is a polar molecule, all the dissolved substances will be polar. Similarly, the dissolved substances out here will also be polar. So if we have polar stuff out here and polar stuff out here, and we have a non-polar barrier, there's no free movement of the particles from inside to outside. Right? So this is how the membrane works. Those non-polar fatty acid residues stop the free movement of polar particles into and out of the cell. So the phospholipids are not the only thing that's in the membrane. There's sugars, there's proteins embedded within the membrane. And those proteins and stuff, they move around in what is known as the fluid mosaic model. Right? So they drift around. Right? And the membrane's fluidity was demonstrated by this experiment that was done. Sorry, that was an accident. Experiment that was done where they took a human cell and they labeled all the proteins of the human cell with a fluorescent dye, and they did the same with the mouse cell, and then they fused the cells together. Now, if those proteins were anchored into place, the dark green would stay on one side and the light green would stay on the other side, right? If, on the other hand, those proteins moved around, you would eventually have a mixture of all the different proteins together, and that is in fact what they observed. Right? So the conclusion here is that the proteins are not anchored in the membrane, but they're free to move around because the membrane is fluid. Right? So you can see here that the membrane, the phospholipid bilayer, is an excellent barrier to the free movement of polar substances. With that being said, you still need to get stuff across the membrane, right? The cell needs some things. It produces waste products that need to go out. There needs to be some sort of traffic into and out of the cell. Now, some things can go through without any issues. Some things are going to have a greater issue. Right? The reason why some things can go through and some things cannot is because the cell membrane exhibits something called selective permeability. Right? Now, the word permeable means that things can go through it. And so the membrane is selectively permeable because some things can go through and other things cannot. It's like your window screen at home. Your window screen is also selectively permeable, right? It's permeable to the air molecules coming in and out, right? But it's not permeable to birds, whatever. In a similar way, the membrane, the phospholipid bilayer, is permeable to some things, but not permeable to other things. So there's 5 basic mechanisms of cellular transport that we'll be covering today. Right? Diffusion, passive transport, active transport, endo and exocytosis. Right, but before we begin, you need to understand a basic rule of nature, and that is particles naturally want to spread themselves out. They want to move from an area of high concentration to an area of low concentration. This process is called diffusion. So if you pour Kool-Aid into a pitcher, all those dye molecules will be concentrated in one spot, but over time they will eventually spread out to maximally occupy the entire pitcher. Right? So they'll spread themselves out until the particles reach equilibrium. Okay, so it's a natural law of things to move from an area of high concentration to an area of low concentration, and that process is called diffusion. So moving down the concentration gradient is what particles want to do anyway. So it doesn't require any energy. It's what particles naturally want to do. Now, some things can actually move down their gradient through the membrane without any issues. Right? So you have to remember that the barrier to the free movement of particles are those nonpolar fatty acid tails. Right? So if you're talking about a molecule that's nonpolar, it can move through without any issues. And you remember how we described nonpolar things? You can't tell one end from the other. So look at things like oxygen. Oxygen is an oxygen atom double bonded to an oxygen. Right? Is this molecule polar or nonpolar? Right? It's nonpolar, right? Because there's no endedness to it. So the concentration on the outside is higher than it is on the inside because the cell is using oxygen, right? So because the concentration is higher on the outside and lower on the inside, and because this molecule is nonpolar, it simply diffuses into the cell. Right? The cell produces CO2. What does CO2 look like? Also a nonpolar molecule. So CO2 builds up inside the cell. The concentration is greater inside the cell than outside. It's a nonpolar molecule, so that molecule simply diffuses out. So oxygen and CO2 can actually diffuse into and out of the cell down their concentration gradient because both of these molecules are nonpolar. Right? Other molecules like glucose, remember glucose is that hexagon-looking thing? That is a polar molecule. Now, the concentration on the inside is low and the concentration on the outside is high. So yes, glucose naturally wants to come in, but it can't because it's polar. So it requires the presence of transport proteins. And through a process called passive transport, the glucose is able to move into the cell through a transport protein. So you can see here we have a transport protein that will allow the movement of glucose into the cell. Now the glucose is moving down its concentration gradient, so this does not require any energy. And this is what we refer to as passive transport. It's passive because no energy is used, but it's transport because it requires the presence of that transporter. The other word for this is facilitated diffusion, right? This is diffusion. It's moving from high concentration to low concentration, but it's being facilitated or helped along by the presence of that protein transporter. Okay? What if you wanted to move things against their concentration gradient? This is going to be a little bit more difficult because particles don't naturally want to move that way. So in order to get the particles against their concentration gradient, you got to give them a little kick in the butt. You got to give them a little bit of energy in the form of ATP. And because you're using energy to move things against their gradients, this is known as active transport. Right, so passive transport, things are moving down their concentration gradient, don't require any energy. Active transport, things are moving against their concentration gradient and do require energy. So here we have H plus ions, protons, right? We have a bunch of protons on the outside, we have fewer on the inside, but we still want to push these guys out. So this is going to require some energy. So we invest some energy, and that transporter will push the protons against their gradients. What if you wanted to bring in something very big or very abundant? Well, you can just simply fold the whole membrane over and create a vesicle like this. This is something called endocytosis. A little invagination will form, and that particle that you want to bring into the cell will get pinched off, and now it is inside the cell in the form of a vesicle. Right? That's endocytosis. Exocytosis is the opposite. You take a vesicle with something that you want to kick out of the cell. That vesicle fuses with the external membrane, and then the particles are expelled. Okay? So those are the basic mechanisms of cellular transport. Now there is one type of cellular transport that I think is really important, and this is called osmosis. And osmosis is the movement of water across a selectively permeable membrane. And the reason why this is important is because it happens in a very predictable way. Right? You should be able to predict what will happen in an osmosis experiment. So here we have a chamber that's separated into two separate containers. Here, one container is separated into two chambers. Right? One side is pure water. The other side is water with some dissolved proteins in it. And the membrane that separates them is selectively permeable. It means that water molecules can go through, but the protein molecules cannot. Right? This is important to understand. You guys realize that if this membrane were permeable to the protein molecules, those green things would simply just spread out across towards the left side, right? But the membrane is not permeable to the green protein molecules. It's only permeable to the water molecules. Okay? So I want to explain osmosis in a couple of ways, three ways. Osmosis is really the movement of water down its concentration gradient. Right? So what's the concentration of water on the left side? It's 100%, right? And on the right side, let's say it's 95%. 95% water, 5% protein. Right? So the concentration of water molecules is higher on the left side than it is on the right side. And like all particles, things want to go from high concentration to low concentration, so the net movement of water will be to the right. Okay? So you can see here that osmosis is really the movement of water down its concentration gradient. However, I don't recommend that you learn it this way, because in order to really understand this, you'd have to really understand in your mind the distinction between concentration and volume. Because we start out by saying there's more water on the left side, right? But then there's movement of water to the right, so you end up with more water on the right. So it can get kind of confusing. So I explain it to you just to illustrate that it is the movement of water. The osmosis is the movement of water down its concentration gradient. Okay? The better way to learn it for you guys is to identify the tonicity of the fluid. Right? And in order to understand how this is going to work, you need to know 3 words, and that is hypotonic, hypertonic, and isotonic. So hypotonic means less dissolved stuff. Remember, those dissolved particles are called solutes. So hypotonic means less dissolved stuff. Hypertonic means more dissolved stuff. And isotonic means same amount. So in this case, the right side is hypertonic. It has more dissolved stuff than the left side, right? So the left side is hypotonic. So once you've identified the tonicity of the solutions, you just need to remember one thing, that water always moves from hypo to hypertonic. So the net movement of water will be to the right. Okay. So a couple of other examples that I can think of. Let's say we have a beaker, and the beaker contains, let's say, a 10% salt solution. And let's say we put a selectively permeable bag that contains, let's say, a 2% salt solution. Alright, so the bag here is selectively permeable. No salt can go through, but water molecules can go through. Alright, so what's going to happen to the bag here? Well, which side is hypotonic and which side is hypertonic? Well, this has more dissolved stuff, so this is hypertonic. And the bag is hypotonic. And because water moves from hypo to hyper, the net movement of water will be out of the bag, and the bag will shrivel up. Okay? Now it's important to note one thing about tonicity. The terms hypo, hyper, and isotonic are relative terms. And what I mean by that is you can't answer a question like, is a 2% salt solution hypo or hypertonic? Right? That's a question that doesn't make sense. Because relative to pure water, a 2% solution is hypertonic. But relative to a 10% salt solution, the 2% solution is hypotonic. Okay? Clear? Now, this becomes really important because when you use terms like hypo or hypertonic, you need to be clear about what side of the water, what side we're talking about. Right? So, using the example that we used before, here we have a beaker, and I said there's a 10% solution, and then the bag has a 2% salt solution, right? So and you guys understand that the net movement of water will be out of the bag. This is the same drawing that I had before. Now, I can ask you I'll ask you a variant of that question. Right? I take a bag containing a hypotonic solution and place it into a beaker. What's going to happen? Well, if this is hypo, then this must be hyper, and the net movement of water will be this way. I can also ask the same question like this. I take a beaker containing a hypertonic solution and place a selectively permeable bag inside. What's going to happen to the bag? Well, if this is hyper, then the bag must be hypo. Same scenario, right? The reason I bring this up, is because it's really important that you understand what I'm asking on a test, right? So, because these terms are relative, I really only have to give you either the bag or the beaker, right? Or I could give you the actual numbers, right? Like in the 10%, 2%. So be really, really mindful of what the questions are asking when you take the exam, okay? So if you take a cell and you place it into an isotonic solution, right, the net movement of water into and out of the cell is about the same. So the cell maintains shape, whether you're talking animals or plants. Right? What about this? If you're placing it into a hypertonic solution, right, the outside solution will be hypertonic, inside solution is hypo. The net movement of water is out of the cell, and the cell shrivels up. If you're a plant cell, the cell wall remains, but the cell will shrivel up. That's something called plasmolysis, by the way. Right. If you place the cells into a hypotonic solution, the cells are hypertonic, and the water will go inside and the cells will swell. All right. So this is a U-tube. Right? It's a U-shaped tube that's separated by a selectively permeable membrane. Right? So you can see here that the two sides of the U have different tonicities. There's more dissolved stuff on the right than on the left. So the right side is hypertonic, the left side is hypotonic. And then through osmosis, the net movement of water will be to the right, and you'll end up with more volume on the right than on the left. Okay? So that's osmosis. It's the movement of water across a selectively permeable membrane. Alright, so we're going to change gears a little bit and talk about another aspect of cell biology, and that is energy. Right? So one of the things that we know about cells is that they all take in and utilize energy in order to survive. So what is energy in the first place? Energy is described by every single book as the capacity to do work, right? And that's, again, like elements. It's not a definition that is immediately clear. What does capacity to do work mean? But rather than belabor the point, right, I think it's one that's it's a word that's very difficult to define, but we all know what energy is, right? Capacity to do work is not really that descriptive a definition, but we all have a sense of what energy is, and that there's different kinds of energy. Okay? So we'll just go with that. The study of energy and the energy transfer between systems is called thermodynamics. And it's governed by 4 laws, but only 2 of them are important for biology. And those 2 are the first law of thermodynamics and the second law of thermodynamics. The first law of thermodynamics, very simple. Energy can be converted from one form to another. Right? And even not knowing anything about biology, you understand that this is how biology works, right? You eat food, you convert the chemical energy into thermal energy to maintain your body temperature, becomes other forms of energy that your body can then use to move your body around, right? So you know that there's energy conversions all over the place, and these are all consistent with the first law of thermodynamics. Now, cells need energy constantly because they're constantly doing stuff, things like active transport, let's say. They're always utilizing energy, and that energy comes from the foods that we eat for us. But ultimately, if we trace back all the energy that we have comes from the foods that we eat. Right? And how did the food get in the energy? Well, if you trace it all the way back, that energy always comes from the sun. Almost always. You sit down to a delicious steak dinner, you're getting biological molecules that your body can use to extract energy from. Why is there energy in your steak? Because your steak came from a cow. Cow walked around. It was a living creature. It clearly had energy. Well, where did the cow get its energy? It got its energy from the grass that it ate. And where did the grass get its energy? It got it from the sun. So you'll remember these terms from the first lecture when we talked in a very abstract manner about the properties of living things. All living things require energy. And the vast bulk of organic energy on Earth comes from the sun. And it's captured by a group of organisms called primary producers. Things that we would call photosynthesizers. And the photosynthesizers are special because they're converting solar energy into glucose, which is a carbohydrate. Yeah, it's a form of chemical energy. And then when organisms eat the plants or eat each other, what they're doing is they're getting energy along the way. Okay? So we can think about the flow of energy from a cellular level and also on an ecosystem level. On an ecosystem level, energy comes from the sun, gets captured by producers, gets eaten by primary consumers, then again, that energy gets eaten by secondary consumers. But the same thing is happening on a cellular level as well. Right? Where a cell, like a plant, is taking sunlight and CO2, turning it into glucose and oxygen, and then the consumers are going to take the glucose and oxygen and turn it back into CO2 and energy. Right? But the energy that's going in photosynthesis is solar. The energy that comes out in respiration is ATP. All right. The second law of thermodynamics states that all things are going to fall apart. That if you don't add any energy to a system, it's simply going to fall apart. And I think this is kind of a not that surprising a conclusion. You guys have experienced this in your own lives. If you don't clean up your room, what happens? It becomes messier and messier and messier and messier. Right? That is the observation that's consistent with the second law of thermodynamics. Right? The reason why your room becomes more and more disorganized is because the energy that's in the room is starting to spread out. Okay? And the only way to maintain order is to add energy into the system. And when you add energy into the system, you can maintain order, right? You have to invest the energy to clean up your room to make it all nice and tidy. But that sort of decay of organization is the second law of thermodynamics. The idea that you don't put any energy in, things will move to a more and more disordered state. That is the second law. So another way of saying this is that the entropy of a system will always increase. Right? The word entropy here refers to disorder. So whereas we originally stated that if you don't add any energy, things become more disordered, right? Said another way, the second law of thermodynamics states that the entropy of a system will increase over time. Okay? So here we have New York City. If you've ever seen those disaster shows like Life After Humans, right? What happens when the humans aren't around to maintain our cities? They simply crumble and fall away, right? And they ultimately get reabsorbed by the earth. Well, what do you think is going to happen to a cell that doesn't receive any energy inputs? It falls apart, and that's consistent with the second law of thermodynamics. Okay? So the term metabolism refers to all the things that are going on inside a living organism. All the chemical reactions that are taking place. Now there are basically two types of chemical reactions. Right? You can divide them into two categories. The ones that build things up and the ones that break things down. The chemical reactions that build things up are called anabolic reactions. And those are always going to require energy. The second class of chemical reactions are called catabolic. And these are chemical reactions that break down things and release energy. So here we have monomers being converted to polymers, right? The polymers are a more complex form. So the conversion of the monomers to the polymers is going to be the anabolic reaction. And that's going to require energy because this is more energetic than the pieces. Breaking them down, that's called catabolism, and energy will be released. So you can couple these anabolic and catabolic reactions to turn one form of energy into another. Right? In the same way that we saw this down here, here we have ATP that gets broken down into ADP in a phosphate. That breakdown is a catabolic reaction that releases energy. Converting ADP and this phosphate back into ATP is an anabolic reaction. So that's going to require energy. Right? And this is going to release energy. You can see here that as we go back and forth, we can turn one form of energy into another form of energy. Right? We can turn our food energy into movement. Right? So the molecule that your cell uses as energy currency is called ATP. It is a type of nucleotide, and it has, you can see, 3 phosphate groups. So in its high energy state, it has, well, let's use this slide. It has 3 phosphate groups here. In its lower energy state, one of the phosphate groups is broken off. It hasn't been consumed, right? Only this bond has been broken. But the third phosphate group has been broken off. So this is adenosine diphosphate with 2 phosphate groups and then a loose phosphate. So even though nothing has been, we've just broken one thing off, this structure right here with the 3 phosphate groups is higher in energy than this structure. It's not because the phosphate is consumed, it's because that piece has been broken off. Right? I want you guys to imagine a pen with a removable cap. Right? The pen with the cap on has higher energy than the pen with the cap off. Even though you haven't eaten the cap, the pen with the cap on is more ordered and more energetic. Okay? In a similar way, having the 3 phosphate groups attached is higher energy than having one broken off. Okay? So you can make these ATP through chemical reactions like cellular respiration, and then we take that ATP and we break it down, and then we use it to run races and do things like that. Okay? Because ATP is a higher energy version of ADP. Okay? So this is the same figure, just turned on its side. Alright? Now, so we know that there are anabolic reactions and there are catabolic reactions. And according to the second law of thermodynamics, everything's going to fall apart, which means that the second law of thermodynamics suggests that catabolic reactions are much more likely to occur over time. That being said, if everything's going to fall apart, why haven't they already? How do we still have high energy compounds? Well, the reason why we have high energy compounds is because they can get stuck in certain high energy configurations. I want you to imagine that you have a mountain. Oops. Get my pen. Right? And if you have a rock up here on this mountain, you can tell with absolute certainty that eventually this is going to end up down here. Okay? How does it stay up there? Why hasn't it rolled down yet? Because there are little jags, like, and maybe your rock is in a little thing like this. So in order to get down, you've got to give it a little bit of push to get over before it falls down. Okay? That little push that you have to give it right there is called the activation energy of a chemical reaction. So high energy molecules can still exist as long as they don't get pushed off the cliff. Okay? Now, living things can make certain chemical reactions happen more frequently than others by using molecules called enzymes. Right? Enzymes are a type of protein that facilitate chemical reactions by reducing the activation energy required to start that chemical reaction. So in our mountain example, you might need, let's say, 30 newtons of force to push this rock out of its little cave or little gully and roll it down the hill. On the other hand, if you've greased the mountain with oil, maybe you don't need 30 newtons anymore. Maybe you can do it with 10 or 12 newtons. That means that rock is much more likely to fall down the hill than it was before the enzyme was there, right? So enzymes make certain chemical reactions happen more frequently by lowering the activation energy required to start the chemical reaction. And they work by literally interacting with the molecules that they are influencing. So for example, there's an enzyme that will break starches up into their monosaccharides. And the way that it does it is it literally grabs the starch molecule and rips it apart. Now, enzymes are proteins, and remember, we've already learned that a protein's shape is what determines its function. So if you can change the shape of the protein, you might change the shape of if you change the shape of the protein, you might change its function, and this includes enzyme activity. And we do know of certain types of enzymes that are influenced by denaturation. There's a reason why a Siamese cat is dark on its face and its ears and its paws and its tail. Those are the coldest parts of the cat's body. In the warmer parts at the core, the temperature is so high that it causes the enzyme to not function, and you're not depositing any pigment in the fur. And that's why the fur of the body is light. Think about a snowshoe hare. Right? There's an enzyme that deposits pigment in the fur, but when it gets to winter time, right, that enzyme changes shape, and the fur that grows then has no color. Right? But it actually turns out to be advantageous for the snowshoe hare because when it's cold, that's when winter's coming, and it's better to be white at that moment, right? But in the springtime, when the snow melts, you don't want to be white anymore. Right? So as the temperature warms up, the enzyme that deposits color into your fur flips back to normal. The fur that you grow is now brown, and you're able to camouflage in the springtime vegetation. Okay? So the point is that enzymes are proteins that can be influenced by all the things that proteins can be influenced by. Right? When we talk about photosynthesis and respiration, there's going to be a long chain of events that occurs, right? Where process 1 gets you product 1, and product 1 goes into process 2 to become product 2, etc. These are what are called metabolic pathways, right? So like assembly lines of molecules, molecular assembly lines. All of these metabolic pathways are mediated by enzymes. And what this means is that your body or cell is very tightly controlling what goes on a chemical level. There's a class of chemical reactions that you need to be aware of, and they're called redox reactions. Redox reactions are ways that cells can transfer energy between molecules. And they do so by either oxidizing or reducing the molecules. So the acronym that you're going to want to remember is oil-rig. Oxidation is loss of electrons, that's oil, and reduction is gain of electrons. So an oxidized compound has had its electrons stripped away from it. and a reduced compound has had electrons thrown upon it. Now, between the oxidized versions and the reduced versions of the molecules are more energetic than the oxidized versions, right? I mean, if you're talking about the same molecule, one version has extra electrons and one has lost those electrons, the one with the electrons is really going to be more energetic than the one without, right? So reduced compounds are the ones that are higher in energy. Now, I told you that redox reactions are ways of transferring energy, right? Because here what you see is that as compound A becomes oxidized, compound B has become reduced, right? So we've donated electrons from one molecule to the other. So what we've done really is we've transferred the energy from A to B. When we talk about photosynthesis and respiration, you're going to see a lot of these molecules throwing electrons around, right, in a series of redox reactions. And this is a way for them to transfer energy from one molecule to the next. Generally speaking, and very commonly, you're going to see these in what are called electron transport chains. Electron transport chains is a series of proteins all lined up in a row. Right? What's going to happen is they're going to pass the electron along this pathway. So as the electron gets passed, this molecule becomes oxidized while the next one becomes reduced. Then as this one gives up its electrons, this one becomes oxidized while the next one becomes reduced. And it's basically like hot potato of electrons. But as they are doing these series of redox reactions, you are able to bleed off the energy of the electrons. I don't know if you guys are aware, but sugar is flammable. Right? If you've ever been camping, just grab a confectioner's sugar, works best. Throw it on the fire, and you'll get this big thing of flame. Okay. Right? That energy that's in the sugar is released all of a sudden, and that's what causes that flame. But you could take that same amount of energy and bleed it off a little bit at a time, and you can actually get more bang for your buck, right? You can actually use the energy that's in the sugar to do stuff like run a marathon or whatever. Okay. So the reason why we want to do these tightly controlled transfers of electrons is so that we can utilize the energy that's in the electrons much more efficiently. Right? And so photosynthesis and respiration is actually, this whole discussion that we've had is really a precursor to a discussion of photosynthesis and respiration. Okay? Where a lot of the principles that we just covered will be revisited and expanded upon.